Good afternoon, everybody. Welcome to the next Infection Immunity Postgraduate Seminar Series. Today we are moving on from bacteria and viruses and onto the world of parasites, which is really great. I'd like to start off by acknowledging the traditional owners of the land and custodians who we meet on today, the Wurundjeri people of the Kulin Nation, and pay my respects to the elders past, present and future. So today we have Sam Emery, who's in the JAX lab. She is going to be doing a great talk today, giving us a bit of hopefully a history and movement of where parasites are and giving us much more idea about what parasites do in our bodies. Sam has done her PhD at Macquarie University. She moved to Weihai in 2016 with a Jack Bracknoff Fellowship. She works on drug resistance, looking at proteomics and really trying to understand from the protein level what these parasites do to our bodies and how they interact with us. So Sam, please give us a talk.
So good afternoon, everyone. I'm Sam, and I'm giving this lecture in lieu of my supervisor, Aaron Jacks, who's currently overseas talking to quite a few of our collaborators in Europe. And within the JAX lab, we work on a very interesting and a very diverse range of parasites, and many of these are gastrointestinal parasites. And what's really important about these parasites is that they are highly successful parasites. There are billions of people around the world who are infected with these parasites, and there are even more people who are at risk of infection. And overall, it is children who are the most at risk. And while there is a great amount of morbidity that is associated with these parasites, we know in children that episodic diarrhea very early in life, under the age of 2 years, is one of the leading causes of mortality. So some of these parasites are really important in terms of decreasing childhood mortality in developing countries. When you have these parasites over a long time chronically, they do lead to cognitive and physical stunting. And overall, these parasites tend to fall under the umbrella of neglected tropical diseases, which means that they have been very underserved by drug discovery, and we have very limited in vitro culture models for the entire parasite in their life cycle, and also very limited genetic and molecular tools in many instances. Why I have named this talk focusing on the complexity of these parasites, that's because they have very complex life cycles. So they are multi-stage. They have sexual and asexual cycles. And what you see in many of these parasites is that they have environmental stages, which facilitates their reinfection and their movement onto the next host. And in the case of the helminth parasites, these stages are crucial for development of these parasites as well. While these are gastrointestinal parasites and the gastrointestinal tract is where these parasites complete their life cycles, they also in some cases have extraintestinal stages, and these can lead to difference in pathology as they move. They also have wide host ranges, so we can see zoonoses from other mammals and vertebrates into humans, and they also have, especially in the case of helminths, very large environmental reservoirs. What's really interesting about this is the complex life cycles means that helminths and protozoans, like all incoming organisms, have evolved mechanisms to maximize their chances of colonization and acceptance in the host in terms of infection. And these generally revolve in strategies around immune evasion, mimicry, and host immunoregulation. So we have very complex interactions between the host and the parasite, between the parasite and the microbiome, and what we are increasingly trying to understand is the way that polymicrobial and polyparasitic infections also interact in each other and how that affects the immune system. So today I'm focusing on the soil-transmitted helminths and the gastrointestinal protozoan. So to start with soil-transmitted helminths, we have 3 main species that infect approximately 1.5 billion people, and approximately 3 times the amount of these people are at risk of these infections. So we're looking at Ascaris, the roundworm, Trichuris whipworm, and hookworm, which is Ancylostoma species and Necator americanus. What's interesting about these species is they have very high associated morbidity, so between 5 to 40 million disability-adjusted life years. But when you consider these organisms, essentially, they don't have very large associated mortality compared with other endemic infections in these areas, including malaria and tuberculosis. Their infection cycles are kind of really involved in terms of chronicity and in reinfection. So because there's such large environmental reservoirs of these organisms within the environment, it means that reinfection is really common. And over time, what happens is you get these parasites reinfecting and establishing patent infection. So these parasites are very long-lived. They can live within the host up to 10 years, and their eggs can remain in the environment for years as well. When it comes to soil-transmitted helminths, the adults don't actually replicate inside the host. What you have is fertile adults and fertile females shedding tens of thousands of eggs per gram of feces into the environment, and this gives that large environmental reservoir. What you also see over time is that the original Th1, Th2 mixed infection, where you have a slightly pro-inflammatory response that is responsible for parasite expulsion, is slowly moved and skewed towards a Th2 response, which is a more regulatory immune response and one of tolerance. So these worms are allowed to stay within the immune system, and they modulate the host immune system in order to facilitate the fact that they won't be expelled. So the commonality between all soil-transmitted helminths is that they have a compulsory life cycle stage within the soil. And within the soil, they undergo embryonation of the egg, and in some cases, they also undergo larval development. So we've got up here the helminths, and you can see the helminths actually undergo several molts from an L1 larvae that hatches out of the egg to an L3. And it's the L3 that actually penetrates the skin, and that's how you get infection with these worms. The pathology and the infection intensity is dependent on how many worms you actually have. When we treat these worms, we treat them with chemotherapeutics known as benzimidazoles, and these facilitate the clearance of the worm, but they don't evolve anything that reestablishes immunity or stops reinfection. So infections can be of varying intensity, and they can be mixed between species. And generally, egg excretion correlates to the number of fertile females that you have within the host, and pathology generally correlates with infection intensity. In terms of the immune response, these are influenced by the route of infection, because if you have oral ingestion versus dermal infection, you're essentially engaging different arms of the immune system in different places. And as mentioned before, while all these worms undergo sexual maturation in the gastrointestinal tract, they do move through extraintestinal stages and tissue migration, and they basically engage and modulate the immune system as they go through these different tissues. As they migrate, they often undergo what are called stage transitions or molts over different larvae, and throughout this time they also engage with host-specific factors, including the microbiome. So in some stages we know that soil-transmitted helminths modulate the composition of the microbiome, and in other cases we also know that the microbiome is essential for development of the worm. Like in some cases in Trichuris murus, the microbiome is responsible for the egg hatching in the cecum. Oops. One thing I want to mention before I move on is essentially there are two types of infection responses. So what we see here is this acute infection, which is a more of the mixed Th1 type response where you get an IgE and a mast cell-based expulsion of the worm. And what you see in chronic infection over time is this more immunoregulatory type response. So over time you have cytokines like interleukin-10 and TGF-beta, which promote a regulatory subset of the immune system called T regulatory cells. And these are responsible for establishing homeostatic interactions in the body, so your tolerance to self-antigens, your tolerance to environmental antigens like your microbiome, and also food antigens as well. So while you have this mixed Th1 response in kind of acute infections, over time with patency and chronic infection, you see a more regulatory type response. So hookworm infects 1 billion people, and this is predominantly via a couple of species, in particular Ancylostoma. You have Ancylostoma duodenale and Selanicum, with Selanicum more prevalent in Asian regions. And you also have Necator americanus. Hookworm is basically responsible for a lot of childhood iron deficiency anemia, and this is a response to the parasite's blood-feeding behaviour. So these worms, as they establish in the intestine, they attach to the mucosa and they feed off the host mucosa and the blood. And one adult ancylostoma duodenale can cause a blood loss in excess of 0.05 mils per worm per day. And you can imagine that if you're infected with up to 100 of these adults, how anemia can quickly establish and how this can have very big impacts on childhood development with iron anemia. It has a complex life cycle. So here we have the compulsory soil stage in here, and during this stage the egg actually hatches, and you can see up here the egg has a very thin wall, so egg hatching happens very quickly, and then we have several molts up to an L3 in the ground, and what happens is these larvae then penetrate the skin, and then they undergo pulmonary migration. So So when they're undergoing pulmonary migration, you can also have transmammary transition. So when you've got the L3 circulating in the blood, they can actually pass to mother and child via breastfeeding as well. So you can see this transition here. After they move through pulmonary migration, they go into the heart and the lungs. And essentially in the lungs, they undergo a third molt to an L4. And then this third, or this L4 is coughed up and then swallowed, where the worm can finally re-establish in the gastrointestinal tract. So the immune response in hookworm is characterized by, as in a lot of helminth infections, this eosinophilia, in particular in the lungs as they migrate and also in the intestines, and elevated IgE. Mast cell degranulation and histamine responses are associated with worm expulsion because these essentially paralyze the worm and they can then be shed out via peristyles. What we see over time is this, essentially in chronic infections, is this immune hyperresponsiveness. So you see that there is less parasite-specific responses, less parasite-specific antigens, and this relates to the fact that we have no age-related immunity. What you also see is induction of interleukin-10, which is really important in terms of regulation of the T regulatory immunosuppressive subset. So in contrast, we have trichuris, which is the whipworm. And trichuris is highly prevalent, and you can shed up to 10,000 eggs per gram of feces if you have a very acute and highly infected. And when you have an acute infection, it's associated with anemia, malnutrition, and the trichuris dysentery syndrome, which is high rates of diarrhea and rectal prolapse. Trichuris infection intensity is very age dependent, and you see children more heavily infected with this parasite. The eggs are very heavily resistant, and you can see up here this very thick wall in contrast to hookworm, so it's very hard to eliminate these eggs from the environment, and they are very long-lived. So it makes it very difficult in endemic regions for you to stop reinfection happening, particularly in children. In contrast to hookworm, we do have this environmental stage here where the eggs are shed and they undergo embryonation and are then ingested. But the entire life cycle of trichuris compared to hookworm, and in contrast to ascaris, is intestinal. So what happens is the L1s invade the villi epithelium, and then they undergo several weeks worth of maturation, and then it takes them a further 3 months to actually mature into a fertile adult. This means that there is quite a delay in egg shedding, which is the main route of diagnosis. So you can actually have about 3 months where you're not shedding eggs, which you aren't diagnosed and you're not being treated. Again, we have eosinophilia that's seen in the intestines, and we also see large amounts of mast cells, of degranulating mast cells, which are associated with the worm being expelled. Elevated IgE is really is another factor that you see, but ultimately in children who are affected with trichuris, histologically they're very dissimilar from uninfected individuals, except very locally around where that worm has penetrated the gastrointestinal tract. And this is believed to be associated with the production of interleukin-10, and we see decreases in interleukin-10 and increases in IgE that are associated with age as children become more immune to this parasite. Lastly, we have Ascaris. So Ascaris is known as roundworm, and in humans, it's caused by Ascaris lumbricoides. And in fact, about 90% of infections are largely asymptomatic. But when you do have acute infection, you see a lot of respiratory-associated symptoms. So clinical signs include coughing, because similar to hookworm, the larvae at one stage are coughed up and swallowed again. So, like Trichuris, Ascaris first develop in the soil for 2 to 4 weeks. And this underoccurs down here, and this is dependent on the environmental conditions. So with good humidity and good temperature, you get faster development. After ingesting the egg, the parasite emerges in the intestines, but then it actually migrates out of the intestines and undergoes pulmonary migration, and it moves into the liver, and you have hepatic migration. This migration through the liver lasts 2 to 4 days, and then the parasite moves into the lungs, where it spends several weeks and it undergoes several molts. So a lot of the development, despite this migration, actually happens within the lungs, and this is where you get this respiratory type responses. The worm is then coughed up, swallowed, and then it undergoes sexual maturation over a couple of weeks in the gut again, in the intestines. We notice with Ascaris, most out of all these worms, a really allergenic type response. And this is particularly what you see with elevated IgE and eosinophilia very much in the lungs. And what we also know is that there's a very strong relation with Ascaris and allergy. So Ascaris is considered a risk factor for asthma, and it is known to exacerbate asthma-type symptoms and also cutaneous-type allergy symptoms from things such as mite bites as well. We know in lab workers who work in it, and we work with Ascaris in our lab, that you actually have to have both respiratory and personal PPE to stop allergy-type responses, because essentially we are seeing you can have induction of allergy-type responses from even just the excretory-secretory products of the worms in an in vitro type environment. So within our lab, as I mentioned, we work on Ascaris, but we actually work on Ascaris sum. So Ascaris sum is a roundworm of pigs, and it has a very similar life cycle compared to the human roundworm, which means that it's a great model for Ascaris because it has the same life cycle, the same tissue migrations, which means you can model the host-parasite interactions and also molecular pathogenesis. And in particular, what we've got here is some of the work that we're trying to do on chemosensation. So as you remember, this parasite moves through the liver over about 2 to 4 days. And to do this, what we're interested in is how the parasite senses and moves through the liver and what it's responding to in the host to make this migration. So on the right here, this is work done by Balu and Pradip. We've got a phylogeny of the emergence of GPCRs. So this is a protein domain that's involved in chemosensation, and it's believed to have emerged around the Cnidaria phylum and then branched off here into the nematodes here. And we're interested in understanding what are the GPCR compositions in these organisms, because we believe these are going to be the chemosensory proteins that are responsible for managing that migration through the liver. So what we've got here is essentially we've been doing hidden Markov modelling. So we've been using C. elegans, which is obviously a model species. It's got really well annotated proteins, and we've taken the GPCR domains out of C. elegans and we've used them to model the GPCR composition within Ascaris. And what's really interesting is that we've got about 1,500 or so of these GPCR domains within about exactly the same 1,500 proteins in C. elegans. And in Ascaris, we've now got about 700 of these GPCR domains within 290 proteins, which means that we have chemosensory proteins that have multi-domain GPCR architecture. So you can see that obviously there's quite a bit of difference between these two organisms in how they can possibly undergo chemosensation. And Pradip in our lab is going to look at actually modelling the migration through the liver via microfluidics. So we're going to prompt larval migration by using specific host metabolites in a microfluidic assay, and that will allow us to understand what these larvae may be responding to. And then we can also map with omics technologies, which of these GPCRs are engaged when we have specific metabolites. So essentially we can arrest migration and allow the immune system to mop up these larvae as they're moving through the liver. To date, though, Aaron has actually done quite a bit of omics work within Ascaris sum over the years. So he was responsible for the draft genome in Ascaris, and he's also done stage-specific transcriptomics. And these are of various larval stages in various migratory states and various genders. So you've got male and female as well down the bottom. And what you can see here, is that there's stage-specific induction of genes in the yellow and the red. So we have stage-specific inductions of genes within the transcriptomes in different larvae, in different migratory stages, and in different genders. When they looked at protein coding genes in the genome, what they noticed was that Ascaris actually had a very large secretome. And in between the secretome, they actually had many peptidases and secreted proteases. And they were able to identify quite a few of these using proteomics in the L3 and L4 larvae. And they could see essentially stage-specific induction of certain proteases in Ascaris sum. And between the genome and the proteome, what they noticed was that there were many orthologs in Ascaris that had been known immunomodulators of various arms of the human immunosystem. So everywhere we've got a red arrow, essentially, is an ortholog that exists in the Ascaris sum genome that has been implicated in immunomodulation of the host. So this leads me into the next section of my talk, which is the immunological hyper-responsiveness that's been observed in individuals infected with helminths. And this is a low antigen-specific response to this helminths, and this response was restored after they've been treated with benzimidazoles, the parasite's been cleared, and this low responsiveness is then lost. So this immunomodulation has been associated over the years with a skewed T cell cytokine profile, and in particular the induction of interleukin-10 and TGF-beta that's responsible for the establishment of a T regulatory cell subset that then essentially suppresses a pro-inflammatory type response. Why this has gained interest over the last 20 or so years is because this response doesn't just extend to the helminths, it extends to what's called bystander antigens. So since about 1968 was the first report that areas that were endemic with helminths also had very low rates of inflammatory diseases. And so what happens is that this suppressive effect, this Th2 skewed profile, then extends to autoimmunity and self-antigens and also environmental antigens as well. Now, this depends on the species. It also depends on infection intensity. If you're highly infected and there is pathology, you're not going to have an immunosuppressive response. And it's also dependent on the species because, as I said, some of these have extraintestinal stages. And so obviously, this immunomodulatory response is dependent on where the worm is located in an anatomical niche. Over time, there's been a concomitant hypothesis known as the hygiene hypothesis, which is the belief that helminth infection early in life is important for the induction of a regulatory type immune system. So while the immune system is maturing, these helminth infections stop autoimmunity and allergy from occurring. And this is the belief that in developing countries, because we've eliminated helminth infections, we have higher rates of these autoimmune and allergy disorders. So over time, people have decided that it's possible in developed countries to use helminths or their products as live helminth therapy to basically treat allergy or pro-inflammatory type diseases, including colitis, Crohn's disease, IBS, and allergies such as celiacs and gluten intolerance. And I've chosen to focus on just a case study today, looking at gastrointestinal helminth infection and multiple sclerosis. Multiple sclerosis is considered a prototypic example of a Th1-mediated organ-specific autoimmunity. And it has defects in both distinct subtypes of regulatory CD4 positive and CD8 positive T cells, as well as a correlation between B cells producing less regulatory interleukin-10. So what I've got here are two studies that were done quite early on in Argentina, basically correlating gastrointestinal nematode infection with repression of disease state in MS. So on the right here, this is from 2007. Essentially, they managed to find 12 patients with clinically remitting relapsing MS, and they were diagnosed with eosinophilia, which is an indication in many cases of gastrointestinal nematodes. They were then shown not to have other gastrointestinal pathogens like protozoans or bacteria or viruses. They were just infected with nematodes. And then these were matched with another clinically remitting relapsing MS patients and healthy controls, 12 each. They then followed these individuals over 5 years, and they looked for degradation of MS via both physical and cognitive testing, and also followed the cytokine profiles in T regulatory populations. And what you can see here on the right was the mapping showed that there was only 3 exacerbations of MS in infected individuals versus 48 in uninfected. So what they were correlating was the lack of a kind of decrease in the MS disease state in the uninfected individuals. And then they also showed down the bottom here that infected individuals had similar numbers of CD4 positive, CD25 positive T cells compared to the controls. And it was only these cells that were essentially expressing FOXP3 transcription factor, which is important for the induction of a regulatory profile in T regulatory cells. In a later study, they also showed that infected individuals also had higher production of B cells producing interleukin-10, which is regulatory. So these were kind of the founding studies that showed in these populations with natural infections that helminth infection was correlated with essentially stopping the progression of MS. And there are subsequent studies that also show that individuals, after they were treated with anthelmintics, they lost a lot of this regulatory functions in their cytokine profiles. There have been a few clinical trials with T. suis ova, so that's Trichuris suis whipworm. It infects pigs, so it has very limited pathology in humans, and it's been used in clinical trials for treatments of MS. Overall, there's actually a very good research climate in Australia for this whole idea of helminth therapy. And I've picked a couple of news stories that are quite spectacular sounding off the ABC website in the last 4 or 5 years. In particular, we have stories here about using hookworm to treat celiacs and gluten intolerance. But I've contrasted these with stories from researchers in Alex Lucas' lab in JCU. And what they've discovered is they've been able to identify the excretory-secretory molecule in liver fluke that promotes wound healing. So in liver fluke, they essentially graze on the liver tissue, and they promote wound healing, so essentially they can replenish their food source. So what they've done is they've actually identified this molecule, and they've taken the live helminth out of helminth therapy. And I think this is really important when understanding this type of therapy, because live helminth therapy is interesting, but it's always going to be much more important to isolate the immunoregulatory molecules, because human responses to helminths are spectral, and it's always going to be a problem to have a helminth therapy with a live worm, because essentially it may have to constantly infect a specific anatomical niche to exert its immunomodulatory effects. We don't know whether this is dose dependent or infection dependent, and we ideally don't want to have to infect these people with worms to maintain an immunomodulatory based effect for the entirety of their lives. So there's some great examples of helminth therapy in the lecture, and I quite like the work that Alex Lucas and his postdoc Michael Smoot have done in essentially taking a molecule out of the liver, and they're going to be able to apply it to non-healing wounds topically in things like diabetes and multiple sclerosis. The other contrast I want to talk about in terms of helminth modulation is the whole idea that bystander antigens also compromises vaccine efficacy, particularly in antenatal vaccination. So while it's great, the idea of helminth immunosuppression in developed countries for helminth therapy, it can compromise one of the greatest technologies we have for allowing us to improve childhood outcomes in terms of many diseases. And what we know is that helminth endemicity overlaps with many diseases like TB, malaria, and HIV, which have much higher incidences of mortality per infection than these helminths do as well. And we've seen correlations with the BCG vaccine for TB, but also for typhoid, measles, and polio. And we believe that part of this may also come from in utero sensitization or tolerization. So mothers who are infected with helminths can pass on some of the tolerization of antigens onto their fetus, and we know this is the case for Ascaris. But also this graph up here shows the generation of IgG, for influenza type B. And what you can see is that there's a lower titer of IgG, which correlates with the number of helminth infections of the mother during pregnancy. So what the model is suggesting down here is that you have helminth infection, which can increase with in utero sensitization, but also once the baby emerges into helminth endemic regions, it's at extreme risk of infection and chronic infection from these helminths. And what you get is this skewed Th2 type profile. What we also don't know, but is obviously playing a very big case, is in these regions with high poverty, you have malnutrition, undernutrition, low protein, calorie, and micronutrient-based diets, which further suppresses the immune system, and these can impair antenatal type vaccines, including BCG. What it's also suggested is that this response can carry on into maturation, which leads to the establishment of a large T regulatory type population, which can further impair Th1 type responses that are needed for clearance of infection in adulthood, such as with TB. So in conclusion to this first half of my talk, soil-transmitted helminths are highly prevalent, they're highly successful, they have very complex but varying and diverse life cycles, but all involve this compulsory life cycle stage within the soil that's crucial for development. And in terms of areas they're endemic, there's a lot of reinfection and there's chronicity, and in a lot of cases, there's, like for hookworm, there's no age-acquired immunity to these infections. We have two types of immune responses down here. We have an acute response, which is an IgE-mediated, mast cell-mediated clearance of the parasite, but then we also have these chronic infections, which are characterized by T regulatory cells and essentially a more regulated immune system. So for the second half of my talk, what I want to focus on is the gastrointestinal protozoan parasites. So I've got three protozoan parasites here on the right. I've got Giardia duodenalis, Entamoeba histolytica, and Cryptosporidium. So Giardia is a very successful parasite with up to 1 billion people around the world infected globally. 300 million of these people go on to develop diarrheal disease known as giardiasis, and this is roughly 500,000 people in Australia. We know that within Australia that it has very high prevalence in children in remote Indigenous communities, and these are often also polyparasitism-based cases with other parasites as well. We know that though Giardia has a very low pro-inflammatory responses, it leads to the induction of many post-infectious clinical sequelae, and these are inflammatory type disorders like IBS and Crohn's disease. Entamoeba histolytica infects around 50 million people, and it's the leading cause of protozoan mortality behind malaria. And this is caused largely by the fact that you can have amoebiasis, which is an extraintestinal pathology of these organisms that moves into the liver, the lungs, and the brain. It also has a very high morbidity, causing roughly over 2 million disability-adjusted life years. Lastly, we have Cryptosporidium, which is a bit different to these organisms here. It has a more complex life cycle with an epicellular stage. It causes diarrheal disease, and it's been implicated as one of the leading causes of mortality in children under the age of 2 years. We know that it infects the immunocompromised, the elderly, and the infants, and in the immunocompromised, they cannot clear this parasite. So usually cryptosporidiosis is self-limiting, but if you do not have a fledged immune system for parasite clearance, you get chronic infection that's life-threatening. And this is particularly a problem because the only drug that we have for Cryptosporidium is not really a parasite-targeted drug. It's an immune adjuvant that essentially allows your immune system to cope with the parasite to expel it. So this drug does not work in a context if you do not have an immune system to actually mount against the parasite. And part of the reason why we've really struggled to control Cryptosporidium and find chemical therapeutics is because we have a limited continuous culture system that allows us to model the parasite and test drugs and therapies. So in terms of the life cycle, both Giardia and Entamoeba are non-invasive. So these parasites do not invade the gastrointestinal tract, they adhere. So you've got Giardia here on the right, and you've got the trophozoites here adhering to the gut epithelial in the villi and the brush border. These parasites, as they move through the gut, they then encyst, and this cyst is environmentally resistant and then can be used to reinfect the next individual via fecal oral route. For Entamoeba, this is pretty much the same. We have another environmentally resistant cyst form and an infectious trophozoite. The difference between Entamoeba and Giardia is that Entamoeba can have this extra intestinal amoebiasis. And that's because the way the trophozoite feeds is it excretes these lytic type cohort of proteases that lyse the cells of the gastrointestinal tract. And what happens is that this lysis can then move through the gastrointestinal lining into the submucosa and facilitate the translocation of the amoeba into the pulmonary system, and then it kind of generally is found in the lungs or the heart, lungs or the brain, or especially the liver, where it causes quite a lot of abscesses. For Cryptosporidium, we have a more complex life cycle. So while this doesn't technically invade the cell, it establishes an epicellular parasitivorous vacuole, which you can see here in the life cycle, which is kind of within the membrane of the cell. So it's not within the cell and it's not extracellular, it's epicellular. The life cycle works by these sporozoites that exist, and then they undergo asexual merogeny here to produce merozoites. These merots then undergo sexual reproduction with microgametes and macrogametes, and then we get oocysts. And what's interesting is, of course, you can get both thin-walled and thick-walled oocysts, and it's the thin-walled oocysts that can propagate autoinfection again and again in chronically infected hosts. We have tried for a very long time to establish culture systems for Cryptosporidium. It has obviously quite a complex life cycle, and many of the cell lines that we have tried, which are from different organisms, only support aspects of this life cycle. So some of them support the asexual cycle, but not the sexual cycle, and even few of them tend to produce oocysts. The other issue is that many of these life cycles are non-replicative. So the amount of oocysts you put in would be the maximal that you would get out. But in most cases, essentially, you're getting less returns for the oocysts that you add into these continuous cell systems. So there's been a lot of effort recently to establish continuous culture systems for Cryptosporidium. And these are just a couple of examples. So at the top here, we have this hollow fiber technology. This is in 2015. Which essentially allows the intestinal epithelial cells to establish in a 3D environment, which is capable of supporting all the stages of the Cryptosporidium life cycle. More recently, we've actually had organoid culture. So we have organoids from the intestines and the lungs, and these can be microinjected with oocysts and sporozoites. And these support all the life cycle stages of Cryptosporidium. And the oocysts that are then taken from this life cycle are infective in vivo. So it looks like a particularly good model. However, the microinjections of oocysts is a bit technical, and we're currently working in the JEX lab to improve cell culture techniques for Cryptosporidium. So what I want to focus on for the last portion of the talk is a parasite that's quite near and dear to my heart. I've worked on Giardia since my PhD, and when you look at its life cycle, it's a fairly simple life cycle, but as a PhD student at our lab, Balu has coined, it lives a short and stressful life. So if you consider parasite success as reinfection, what Giardia has to do is it has to exist, then it has to replicate and establish infection in the intestines, and then also generate cysts to reinfect the next host. To do this, Giardia has to cope with physiological, xenobiological, and environmental stresses along the way. And what I want to focus in particular is this physiological stress. So this is innate and specific immune responses from the human host that try to essentially stop Giardia from establishing and replicating. And this has led to quite complex host-parasite interactions in Giardia. And what we're starting to understand, most interestingly, is the crucial role that the microbiome plays in this interaction in Giardia. So when we consider this kind of triad of host, parasite, and the microbiome, We know essentially that Giardia interacts with the host to establish infection, so to adhere to the gut epithelial, to make it through the host barriers and to establish and proliferate. In interacting with the host and the host barriers, the host engages a wide range of innate and specific immune responses, defensins, nitric oxide, and pro-inflammatory cytokines. And what we've started to understand in the last 5 or so years is that the induction of Giardia's secretome is responsible for degrading these proteins and essentially stopping its expulsion via the immune system. More recently and interestingly, we understand that Giardia's interactions with the microbiome are not particularly good for the host. So we understand that Giardia not only makes the gut a more advantageous place for colonizations of enteropathogens like Salmonella and E. coli, but the interactions that Giardia has with the commensal bacteria essentially produce pathobionts. So these are kind of induction of gene clusters and phenotypes of the microbiota that can then perpetuate pro-inflammatory responses in the host long after the parasite is cleared. And so what we're starting to understand is that the post-infectious sequelae in Giardia is probably actually mediated by its interactions with the microbiome more so than with the host. We also know that the microbiome in Giardia can actually promote Giardia being cleared from the infection tract or never being established. So we know in mice that are sharing kennels, in sharing their commensal bacteria, there are some flora of the microbiome that's protective against giardiasis. And there's also been a lot of work recently to understand the role of lactobacillus in antagonizing Giardia infection and preventing Giardia from establishing. What we understand very little about is co-infection and polymicrobial interactions between Giardia. We understand that Giardia can in some cases protect against other diarrheal diseases, but we also know that enteropathogens can be immunomodulated, can have the host impacts immunomodulated by Giardia concurrently. We also know that if helminths change the microbiome, that in some cases they may also promote microbiome interactions that are less advantageous for Giardia. So we know in some cases lactobacillus is correlated with some gastrointestinal nematodes, and Giardia doesn't have particularly positive interactions with them. What we understand even less about is the role of subspecies and isolate variation and how we could have isolate-specific virulence in Giardia. And there's recently having a look at Giardia's metabolomes and how different isolates and different strains may have different metabolic requirements that may allow them to establish more virulent infections. So in terms of Giardia's gut interactions, it's very important to remember that Giardia is non-invasive. So at no stage is it ever invading the gut epithelial. It's attaching and interacting. To establish infection, Giardia must attach to the microvilli. And to do this, it must breach several gut and microbiomes to do so. And what's interesting is that infection is characterized by the lack of an inflammatory response, whereas post-infection we see an increased inflammatory response, which is suggestive of immunomodulation. So what I want to focus is the secretome's role in pathogenesis in Giardia, and particularly both cathepsin B-like cysteine proteases, but also the way Giardia uses its arginine catabolism to actually immunoregulate the host. Towards the end of my PhD, I showed that Giardia's secretome can be induced without attachment, that Giardia starts secreting proteases as soon as it senses any host factors, including the excretory-secretory products. And what we know in pathogenesis is that the secretome is essential for overcoming the host barriers in the form of the microbiota layers, which tends to occur in biofilms over the gut epithelial, for degrading the mucin, which occurs over the epithelial before that prevents attachment, and also in changing and remodeling the epithelia, including the villen border, the brush border, and the apical junctions down the bottom here. So in terms of cysteine proteases, I want to focus on several interactions that have occurred and that have been shown experimentally to be regulated by proteases. So these include, firstly, the degradation of host mucin by Giardia's proteases, then the degradation of villen, which is an actin modeling protein on the surface that is crucial for the brush border and the villi. the disruption of the apical junction complexes and tight junctions between these cells, and then the disruption of the commensal homeostasis, which is partly mediated by the disruption of the biofilms and the mucin barrier, but also it appears that Giardia also disrupts the homeostatic gene expression profiles of commensals and induces pathobience of them. And then lastly, we know Giardia directly degrades pro-inflammatory cytokines to mediate the pro-inflammatory response and to stop the migration of pro-inflammatory cells. And many of these effects are blocked in vitro with protease inhibitors, which I'm going to show in the next couple of slides. So the first barrier is the mucin layer over the cells. And we've got up the top here some colon biopsy specimens from humans that have been co-incubated with Giardia. And the yellow line shows essentially the mucin layers. And you can see here they've measured on the graph here that Giardia during co-incubation of only an hour and a half starts to degrade the mucin layer. What they also show in vitro models is that in globulate cells, that within about an hour and a half, Giardia rapidly induces mucin transcription in host cells. And if you treat the Giardia with a protease inhibitor, you don't get degradation of mucin and you don't get induction of this mucin transcription either. So what was interesting was these were done in vitro, but they were also able to then show that this was occurring in vivo in mouse models. So we've got here mouse models with a knockout of the mucin-2 gene, and you can see that the knockout of the mouse had both higher levels of trophozoite colonization, but also had a weight impairment. And weight impairment or failure to thrive is a very important pathology that's seen in children infected with Giardia. What they also saw was after degradation of the mucin, they also had translocation of bacteria in the gut across the barrier, which you can see here with these arrows and these green fluorescents, and they could detect higher levels of these bacteria both in the spleen and the liver. And then recently what we were also shown was that several of the cysteine proteases in Giardia actually had specificity in terms of their amino acid targets within sequences for cleaving mucin 2, mucin 4, and mucin 16. So the excretory-secretory system in Giardia is responsible for this degradation of the mucin layer that allows the establishment of the trophozoite, and then also down the road may also be responsible for translocation of commensals. The next layer of the gut barrier involves both the brush border, which is mediated in many ways by vilin. Vilin is a cytoskeletal protein of the gastrointestinal epithelial. It regulates the brush border, and it's also responsible for regulating the formation and the organization of actin at this brush border. It's also a pro-survival factor, so depletion of vilin can also signal through apoptosis, and Giardia is known to trigger apoptosis very early in these interactions. So what they showed was that Giardia excretory-secretory products degraded vilin. So we've got this 45 kilodalton degradation band here, and that we could stop this degradation with treatment with protease inhibitors. What was really interesting was that this degradation of vilin was dependent on Giardia contacting host cells. So if you put Giardia in a trans well, when the trophozoites can't actually get to the epithelial, they don't degrade vilin. And this suggests that it's actually a surface-bound protease on the parasite surface that's mediating this response. And we can see up here the degradation of the vilin around here in the lack of fluorescence. Once you actually move through the vilin, there's the apical junction complex, and this is regulated by a host of proteins, including claudins, oculins, and actin fibers. And what they've shown recently is that Giardia doesn't necessarily change the expression of these proteins, but it causes a reorganization of them. So spatially, these proteins should be expressed around the surface of the cell at these junctions, but when Giardia co-incubated with these layers, we essentially start to see these proteins internalized. And this matched up really neatly with a recent experiment that came out from researchers, which characterized Giardia pain, which is a papain-like cysteine protease that has a fully intact catalytical triad. And what they showed is that you've got the intestinal epithelial here, which is all nicely contacted. When you incubate with just Giardia pain, you start to see the breakdown of these apical junctions and then induction of apoptosis in these blebbing profiles here. Again, what you can see here on the right is that this protease alone is enough to induce the internalization of many of the apical junction proteins, which is important for maintaining these cell-to-cell contacts that you see up here. Lastly, what they also saw was that this protease alone was pro-apoptotic, so they had induction of caspase 3 here, and they had cleavage of PARP, which was fully processed within 2 hours. So this protease is important in regulating Giardia's pathogenesis in breaking down these cell-to-cell contacts and the cell epithelial. As mentioned before, because Giardia disrupts these cell layers, they change the gut microbiome and the organization and also the phenotype of the microbiome. So we know that Giardia changes the composition of microbiome samples that are established from patients from healthy samples up here. And you can see the increase in particularly Clostridial type bacteria. We also know that Giardia degrades the extracellular matrix, and this breaks down the biofilm composition which occurs over the epithelial, which you can see up here. What happens is that these bacteria no longer occur in a biofilm type organization, they become planktonic, which means that they're swimming around and they're motile, and they have a much higher chance of translocating as the apical junctions are broken down and inducing inflammation submucosally. What was interesting was in this study here was they showed that many of these planktonic swimming bacteria, even once the Giardia were removed, were still capable of stimulating pro-inflammatory responses. So this comes back to the idea that commensal bacteria then become pathobionts that even in the absence of Giardia can continue to propagate pro-inflammatory responses after parasite clearance. And this is believed to be occurring via Giardia's induction of synthetic gene clusters. So there was an experiment done where Giardia and E. coli were incubated with C. elegans. And when Giardia and E. coli were co-incubated, there was a decrease in the survival of the C. elegans. And what they showed transcriptionally was that exposure of the E. coli to Giardia turned on hydrogen sulfide type responses, which were then responsible for killing the worm. So you have this shift in both the phenotype and the expression profile of commensal organisms, which can turn them into pathobionts long after the parasite is gone. So the last kind of immunomodulatory effect of proteases that I want to talk about is essentially the degradation of actual cytokines from the immune system. So in 2005, we were starting to get transcriptomics that was indicating that Giardia was greatly upregulating many pro-inflammatory cytokine transcripts, but these did not match or correlate with the protein levels that were taken from the media. And what we've got up here is essentially you can see Giardia is degrading interleukin-8 and degrading several other cytokines through here, and these are responsible for kind of post-translational repression of the host immune system. This matched up to several studies that were done in about 2014, which showed that the secretory products of Giardia were essentially degrading interleukin-8, and they could do this degradation in the presence of stimulation of interleukin-8 and also in samples taken from patients that had pro-inflammatory type disorders like Crohn's disease. So they were highly immune suppressive. And then lastly, down here, what you can also see is that this effect was stopped when they were treated with protease inhibitors, which further implicated the role of the secretome in this virulence. So lastly, in repressing pro-inflammatory responses like interleukin-8, Giardia makes the gut more advantageous for enteropathogens. So this attenuation of interleukin-8 could be done in the presence of Salmonella typhimurium toxin. So it induces interleukin-8, but what you can see up here is that essentially Giardia was attenuating the Salmonella induction of interleukin-8, and this was done without actually affecting the viability of the Salmonella. So in this way, Giardia's host immunomodulation can extend to other enteropathogens. This was also shown in a model of bacterial colitis. So they induced colitis using Clostridium toxins, which are known to cause diarrhea. And basically, Giardia reduced levels of pro-inflammatory mediators, and you can see down here that we're getting less induction of this yellow, which is associated with granular site infiltration in Giardia. But what was interesting is down here, this effect was isolate dependent. So we have one isolate here, which is clearly reducing this effect, but another isolate here, which was not. So this may be responsible for isolate variation and isolate variation in pathogenesis. And lastly, really briefly, just to change direction, one of the ways that Giardia actually mediates host immune response is using its arginine catabolism. So Giardia uses arginine for energy generation, and it rapidly eats the host for arginine within the intestine. This is really beneficial for Giardia because if the host is deprived of arginine, it cannot produce nitric oxide. Nitric oxide is essentially very bad news for Giardia. What you can see here is that essentially it's cytostatic, it stops Giardia from proliferating, and it affects their adherence, so they can be cleared via peristalsis. So Giardia not only secretes its arginine catabolism enzymes, it rapidly takes up arginine via an antiporter for orthonin and blocks the host from getting arginine and producing nitric oxide. And by doing this, the host cells are unable to proliferate and eventually undergo apoptosis. So in conclusion, with Giardia, you can see that pathogenesis is highly dependent on interaction with the host epithelial and the gut structure, the mucin layer, the villan layer, brush borders, and cell junctions. During this establishment, Giardia interacts with the microbiome, and it changes the phenotype of the microbiome from a largely biofilm-like organisation into swimming planktonic bacteria that then become pathobionts that can stimulate pro-inflammatory responses. We also see that Giardia directly interacts and immunomodulates the host through degrading pro-inflammatory cytokines and through blocking nitric oxide production. So with that, I'd like to thank some of Aaron's collaborators on his omics of gastrointestinal nematodes, and some of my lab mates as well who have been involved in some of the work in this talk. And yeah.
You mentioned inhibitors for the proteases, which are Giardia. What are they?
What are the inhibitors? It's generally E64. They're very broad inhibitors. They've tried more specific inhibitors of cathepsin B, and they don't inhibit the effect. So essentially only very broad inhibitors of cysteine proteases block these effects, and it's probably due to the fact that there's very limited homology. So the inhibitors we have are only very broad. And that's the real limitation of a lot of these talks, or a lot of these experiments, is because we're using such broad inhibitors, we can't get to the very precise protease that's mediating this, because essentially when we inhibit them, we're knocking out all the secretory-based products. So we don't know in many cases what's the specific protease that's mediating these interactions.
Good talk. I was just wondering, breaking down the tight junctions with Giardia, how does that increase its survivability, and why did evolutionary go for that?
In terms of the apical junctions, it is a problem because essentially it allows for antigens to start to move through into like the basal layers, which can stimulate a lot of T cell based interactions. I think in many occasions it's the damage to the epithelial that kind of mediates a lot of the pathogenesis, and in some cases you don't have real induction of apoptosis. It's only in more virulent strains, and it may be that, as you kind of have pointed out, that apical junctions are not particularly useful for degrading, but the broad specificity of these proteases and the fact that they do need to degrade the top of the layer, they really need to reduce the brush border in order to get down to attach. So it may be that some of the surface proteins in particular at that brush border are more of the targets than the junctions.
Thank you, Sam. For your time today. Thank you very much.